BACE Practice Test 2026 Biotechnician Assistant Credentialing Exam All You Need to Know 2026. Question 1. After performing the polymerase chain reaction with the primers she designed to amplify LON A technician found that her reaction contained two distinct bands, one at the expected size of 1,200 kilobase pairs, and another unexpected band at 2,000 kilobase pairs. What is the most likely reason there is an unexpected band in her reaction? Option A. She used an annealing temperature that was too low. Option B. She used an extension time that was too long. Option C. She used a denaturation temperature that was too high. Option D. She used too much DNA polymerase. The correct answer is option A. She used an annealing temperature that was too low. An annealing temperature that is too low allows primers to bind to nonspecific sequences on the DNA template, resulting in the amplification of unintended products. Question 2. In a biotechnology laboratory, tap water is best used for which of the following tasks? Option A. The first rinse when washing glassware. Option B. Preparing final phosphate buffers. Option C. Making cell culture media. Option D. Final rinsing of microcentrifuge tubes. The correct answer is option A. The first rinse when washing glassware. Tap water contains minerals and impurities that can interfere with experiments, so it is only suitable for the initial rinse during glassware cleaning. Question 3. The order of amino acids on a polypeptide chain is encoded by which molecule? Option A. Transfer RNA. Option B. Messenger RNA. Option C. Ribosomal RNA. Option D. Small nuclear RNA. The correct answer is option B. Messenger RNA. Messenger RNA carries the genetic information from the DNA in the nucleus to the ribosome, where the sequence of codons determines the order of amino acids. Question 4. How do deoxyribonucleic acid molecules vary from one species to another? Option A. The type of phosphate group used. Option B. The structure of the sugar backbone. Option C. The number of genes and non-coding regions. Option D. The direction of the double helix twist. The correct answer is option C. The number of genes and non-coding regions. While the basic structure of DNA is universal, species differ in the total number of genes, the specific sequences of those genes, and the amount of non-coding RNA. Question 5. What is agar? Option A. A liquid broth used for cell suspension. Option B. A chemical used to stain DNA. Option C. A detergent used to lyse cells. Option D. Solid media on which bacteria grow. The correct answer is option D. Solid media on which bacteria grow. Agar is a gelatinous substance derived from seaweed that is used as a solidifying agent in culture media for growing microorganisms. Question 6. What part of one amino acid differs from another amino acid? Option A. The amino group. Option B. The carboxyl group. Option C. The R group. Option D. The alpha carbon. The correct answer is option C. The R group. Each of the 20 amino acids has a unique side chain called an R group, which determines its specific chemical properties. Question 7. How are the proteins and fractions collected during column chromatography visualized? Option A. Using a simple visual inspection of color. Option B. Using polyacrylamide gel electrophoresis. Option C. Using a standard light microscope. Option D. Using a high-speed centrifuge. The correct answer is option B. Using polyacrylamide gel electrophoresis. Sodium dodecyl sulfate polyacrylamide gel electrophoresis is used to separate and visualize proteins based on their molecular weight to confirm which fractions contain the target protein. Question 8. Which of the following is a messenger RNA codon? Option A. TAT. Option B. UAU. Option C. ATG. Option D. GCG. The correct answer is option B. UAU. Messenger RNA uses the nitrogenous base uracil instead of thymine, so UAU is a valid codon while those containing T are not. Question 9. What is the purpose of sodium dodecyl sulfate when used in polyacrylamide gel electrophoresis? Option A. To increase the speed of the current. Option B. To stain the proteins for visualization. Option C. To bind to the DNA backbone. Option D. To denature the proteins and give them a negative charge. The correct answer is option D. To denature the proteins and give them a negative charge. Sodium dodecyl sulfate is an anionic detergent that unfolds proteins and coats them with a negative charge so they migrate through the gel based strictly on size. Question 10. Which department is in charge of testing a biopharmaceutical product to ensure its safety and potency? Option A. Research and Development. Option B. Manufacturing. Option C. Quality Control. Option D. Human Resources. The correct answer is option C. Quality Control. Quality control is responsible for the analytical testing of raw materials and finished products to ensure they meet established specifications. Question 11. Lipids provide which of the following functions for cells? Option A. Primary source of genetic information. Option B. Stored energy. Option C. Catalyzing chemical reactions. Option D. Transporting oxygen in the blood. The correct answer is option B. Stored energy. Lipids like fats and oils serve as long-term energy storage molecules and are also essential components of cell membranes. Question 12. Which of the following is a monosaccharide commonly used as an energy source for cells in cell culture media? Option A. Sucrose. Option B. Lactose. Option C. Glucose. Option D. Starch. The correct answer is option C. Glucose. Glucose is a simple sugar that cells readily take up and metabolize through glycolysis to produce adenosine triphosphate. Question 13. Where does translation occur in prokaryotic cells? Option A. Inside the nucleus. Option B. In the mitochondria. Option C. At the ribosomes. Option D. Within the endoplasmic reticulum. The correct answer is option C. At the ribosomes. Since prokaryotes lack a nucleus, translation of messenger RNA into protein occurs on ribosomes located in the cytoplasm. Question 14. In aerobic respiration, carbohydrates are ultimately broken down into which of the following? Option A. Carbon dioxide and water and adenosine triphosphate. Option B. Lactic acid and ethyl alcohol. Option C. Glucose and oxygen. Option D. Carbon monoxide and hydrogen. The correct answer is option A. Carbon dioxide and water and adenosine triphosphate. Aerobic respiration uses oxygen to fully oxidize glucose, resulting in the production of carbon dioxide, water, and usable energy in the form of adenosine triphosphate. Question 15. Most adenosine triphosphate in eukaryotic cells is produced in which organelle? Option A. The Golgi apparatus. Option B. The mitochondria. Option C. The lysosomes. Option D. The chloroplasts. The correct answer is option B. The mitochondria. Mitochondria are known as the powerhouse of the cell because they are the site of the electron transport chain and oxidative phosphorylation. Question 16. What is the definition of an aqueous solution that yields a hydrogen ion when ionized? Option A. Base. Option B. Buffer. Option C. Acid. Option D. Salt. The correct answer is option C. Acid. An acid is a substance that increases the concentration of hydrogen ions when dissolved in water. Question 17. What is defined as the moles of solute per liter of solution? Option A. Molility. Option B. Molarity. Option C. Normality. Option D. Mass percent. The correct answer is option D. Mass percent. Molarity is a common unit of concentration in chemistry representing the amount of a substance in a specific volume of solution. Question 18. What is a solution that resists changes in pH when small amounts of acid or base are added? Option A, solvent. Option B, catalyst. Option C, buffer. Option D, suspension. The correct answer is option C, buffer. Buffers are composed of a weak acid and its conjugate base, which work together to neutralize added hydrogen or hydroxide ions. Question 19. What is a measurement of the concentration of hydrogen ions in a sample? Option A, molarity. Option B, absorbance. Option C, temperature. Option D, pH. The correct answer is option D, pH. The pH scale is a logarithmic measure of the acidity or alkalinity of a solution based on hydrogen ion activity. Question 20. What type of test is used to measure the amount of total protein present in a sample? Option A, potency assays. Option B, activity assays. Option C, concentration assays. Option D, sterility assays. The correct answer is option C, concentration assays. Concentration assays determine the total quantity of protein in a sample, often using methods like the Bradford or BCA assay. Question 21. What type of test is used to measure the amount of active ingredient in a drug? Option A, potency assays. Option B, loading assays. Option C, stability assays. Option D, identity assays. The correct answer is option A, potency assays. Potency assays are specifically designed to measure the biological activity or strength of the active pharmaceutical ingredient. Question 22. What type of test demonstrates whether or not a specific enzyme or drug is having the desired effect for which it was designed? Option A, concentration assays. Option B, activity assays. Option C, purity assays. Option D, endotoxin assays. The correct answer is option B, activity assays. Activity assays evaluate the functional performance of a molecule to ensure it performs its intended biological task. Question 23. What is a highly specific test to determine protein concentration based on a particular antigen and antibody interaction? Option A, polymerase chain reaction. Option B, mass spectrometry. Option C, enzyme-linked immunosorbent assay. Option D, nuclear magnetic resonance. The correct answer is option C, enzyme-linked immunosorbent assay. An enzyme-linked immunosorbent assay uses the specificity of antibodies to detect and quantify specific proteins in a complex mixture. Question 24. What is a document listing the detailed steps of key processes to help ensure consistent and high-quality output? Option A, batch record. Option B, material safety data sheet. Option C, standard operating procedure. Option D, product label. The correct answer is option C, standard operating procedure. Standard operating procedures provide standardized instructions to ensure tasks are performed correctly and consistently every time. Question 25. What is the viscous buffer used to track DNA or proteins during electrophoresis? Option A, ethadium bromide. Option B, loading dye. Option C, running buffer. Option D, restriction enzyme. The correct answer is option D, restriction enzyme. Loading dye adds density to the sample so it sinks into the well and provides a visible color front to monitor the progress of the run. Question 26. Which document used in biomanufacturing contains step-by-step instructions and spaces to record information during the production process? Option A, the laboratory notebook. Option B, the standard operating procedure. Option C, the batch record. Option D, the certificate of analysis. The correct answer is option C, the batch record. A batch record serves as a legal document that tracks every step taken and every measurement made during the production of a specific lot of product. Question 27. What is the quantitative measurement of absorbance based on visible and ultraviolet light sources passing through a solution? Option A, spectrophotometry. Option B, chromatography. Option C, centrifugation. Option D, microscopy. The correct answer is option B, chromatography. Spectrophotometry measures how much light a chemical substance absorbs by determining the intensity of light as a beam passes through the sample solution. Question 28. What is the use of an electric field to separate DNA in a porous matrix? Option A, osmosis. Option B, gel electrophoresis. Option C, diffusion. Option D, active transport. The correct answer is option C, diffusion. Gel electrophoresis separates molecules like DNA based on their size and charge as they move through an agarose or acrylamide gel. Question 29. What is the process of making RNA from a DNA template? Option A, translation. Option B, replication. Option C, transcription. Option D, transformation. The correct answer is option C, transcription. During transcription, the enzyme RNA polymerase copies a specific sequence of DNA into a complementary strand of messenger RNA. Question 30. What is the process by which messenger RNA is decoded and polypeptides are formed? Option A, transcription. Option B, transduction. Option C, translation. Option D, transfection. The correct answer is option C, translation. Translation is the cellular process where ribosomes synthesize proteins using the genetic code carried by messenger RNA. Question 31. What are autonomously replicating small circles of DNA present in some bacteria? Option A, chromosomes. Option B, plasmids. Option C, ribosomes. Option D, centrioles. The correct answer is option B, plasmids. Plasmids are extrachromosomal DNA molecules often used in biotechnology as vectors to carry and clone specific genes. Question 32. What is the deliberate modification of the characteristics of an organism by manipulating its genetic material? Option A, natural selection. Option B, genetic engineering. Option C, traditional breeding. Option D, spontaneous mutation. The correct answer is option C, traditional breeding. Genetic engineering involves using recombinant DNA technology to alter the genetic makeup of an organism for a specific purpose. Question 33. Parkinson's disease, diabetes, and cystic fibrosis are examples of which type of condition? Option A, infectious diseases. Option B, genetic disorders. Option C, nutritional deficiencies. Option D, environmental allergies. The correct answer is option B, genetic disorders. Genetic disorders are caused by abnormalities in an individual's DNA, which can be inherited or occur spontaneously. Question 34. Which type of organism does not have a nucleus or membrane-bound organelles, so its DNA is contained in the cytoplasm? Option A, eukaryote. Option B, prokaryote. Option C, fungi. Option D, protist. The correct answer is option A, eukaryote. Prokaryotes, such as bacteria, have a simpler cell structure where genetic material is not sequestered within a nuclear envelope. Question 35. Which type of organism contains a nucleus and other organelles enclosed within membranes? Option A, prokaryote. Option B, eukaryote. Option C, virus. Option D, prion. The correct answer is option B, eukaryote. Eukaryotes include animals, plants, and fungi, characterized by complex internal compartmentalization and a defined nucleus. Question 36. What is the process of correcting mutations which cause genetic diseases by delivering therapeutic DNA into a patient's cells? Option A, chemotherapy. Option B, gene therapy. Option C, antibiotic therapy. Option D, vaccination. The correct answer is option B, gene therapy. Gene therapy aims to treat or prevent disease by inserting a functional gene into a patient's cells to replace a missing or defective one. Question 37. What is the use of living systems or organisms to develop or make useful products? Option A, biochemistry. Option B, biotechnology. Option C, biophysics. Option D, biogeography. The correct answer is option D, biogeography. Biotechnology encompasses a wide range of procedures for modifying living organisms according to human purposes. Question 38. What is the deliberate linkage of DNA from different sources into one molecule? Option A, genomic DNA. Option B, recombinant DNA. Option C, complementary DNA. Option D, mitochondrial DNA. The correct answer is option B, recombinant DNA. Recombinant DNA molecules are formed by laboratory methods of genetic recombination to bring together genetic material from multiple sources. Question 39. What is the formula for the dilution of stock solutions? Option A, mass 1 times volume 1 equals mass 2 times volume 2. Option B, concentration 1 times volume 1 equals concentration 2 times volume 2. Option C, concentration 1 plus volume 1 equals concentration 2 plus volume 2. Option D, pressure 1 times volume 1 equals pressure 2 times volume 2. The correct answer is option B, concentration 1 times volume 1 equals concentration 2 times volume 2. The formula concentration 1 times volume 1 equals concentration 2 times volume 2 is used to determine how much concentrated stock solution is needed to make a specific volume of a more dilute solution. Question 40. True or false? Cells produced by mitosis are haploid. Option A, true. Option B, false. The correct answer is option B, false. False. Mitosis produces two genetically identical daughter cells that are diploid, meaning they have the full number of chromosomes. Question 41. True or false? Germinating or sprouting plants are actively undergoing mitosis. Option A, true. Option B, false. The correct answer is option A, true. True. Germination involves rapid cell division and growth as the plant embryo develops into a seedling. Question 42. True or false? Meiosis occurs in all sexually reproducing eukaryotes. Option A, true. Option B, false. The correct answer is option A, true. True. Meiosis is the specialized cell division process that produces gametes with half the chromosome number for sexual reproduction. Question 43. True or false? Meiosis is a type of cell division which increases the chromosome number by 2. Option A, true. Option B, false. The correct answer is option B, false. False. Meiosis actually reduces the chromosome number by half to ensure that the offspring have the correct number of chromosomes after fertilization. Question 44. True or false? During meiosis, chromosomes may cross over and shuffle their genetic information. Option A, true. Option B, false. The correct answer is option A, true. True. Crossing over occurs during prophase 1 of meiosis and increases genetic variation in the resulting gametes. Question 45. True or false? Plant breeders can take advantage of meiosis by selective breeding where pollen from plants with desired traits are purposely crossed with other plants. Option A, true. Option B, false. The correct answer is option A, true. True. Selective breeding utilizes the natural process of meiosis and fertilization to combine beneficial traits in new plant varieties. Question 46. True or false? DNA replication occurs in the 3' to 5' direction. Option A, true. Option B, false. The correct answer is option B, false. False. DNA polymerase can only add nucleotides to the 3' end of a growing strand, meaning replication occurs in the 5' to 3' direction. Question 47. True or false? DNA polymerases are the enzymes that create DNA molecules by assembling nucleotides. Option A, true. Option B, false. The correct answer is option A, True. True. DNA polymerase is the primary enzyme responsible for synthesizing new DNA strands during replication. Question 48. True or false, DNA polymerases from thermophiles or heat-loving bacteria are used in the polymerase chain reaction? Option A, true. Option B, false. The correct answer is option A, true. True. Heat-stable polymerases like TEC are essential for the polymerase chain reaction because they can withstand the high temperatures used to denature DNA. Question 49. True or false, DNA is replicated on the leading and the lagging strands in exactly the same way? Option A, true. Option B, false. The correct answer is option B, false. False. The leading strand is synthesized continuously, while the lagging strand is synthesized discontinuously in short segments called Okazaki fragments. Question 50. True or false, the Food and Drug Administration requires biopharmaceutical companies to follow current good manufacturing practices? Option A, true. Option B, false. The correct answer is option A, true. True. Current good manufacturing practices are regulations that ensure the quality, safety, and purity of pharmaceutical products. Question 51. Which of the following is not a document used in the manufacture of a biopharmaceutical product? Option A, batch record. Option B, standard operating procedure. Option C, batch recipe. Option D, material safety data sheet. The correct answer is option C, batch recipe. While recipes exist, the formal controlled documents used in manufacturing are the batch record and the standard operating procedure. Question 52. Which department in a biopharmaceutical company has the final say if a production batch may be released for sale or use? Option A, manufacturing. Option B, quality assurance. Option C, marketing. Option D, research and development. The correct answer is option B, quality assurance. Quality assurance reviews all production and testing records to ensure every step met the required standards before a product is released. Question 53. Which department in a biopharmaceutical company is responsible for testing a product to ensure it is safe and efficacious? Option A, quality control. Option B, logistics. Option C, finance. Option D, public relations. The correct answer is option A, quality control. Quality control performs the physical, chemical, and biological tests required to verify the safety and efficacy of the product. Question 54. Which department in a biopharmaceutical company is responsible for identifying future products? Option A, human resources. Option B, legal. Option C, research and development. Option D, facilities management. The correct answer is option D, facilities management. Research and development focuses on the discovery of new drugs and the improvement of existing technologies. Question 55. True or false, the study of decision-making as it applies to moral decisions regarding advances in biotechnology is called bioethics? Option A, true. Option B, false. The correct answer is option A, true. True. Bioethics addresses the ethical implications and social concerns arising from new biological discoveries and technologies. Question 56. Food and Drug Administration regulations for the biopharmaceutical industry can be found in which publication? Option A, the Merck Index. Option B, the Code of Federal Regulations. Option C, the Journal of Biological Chemistry. Option D, the Physician's Desk Reference. The correct answer is option B, the Code of Federal Regulations. The Code of Federal Regulations contains the official and permanent rules established by the federal government. Question 57. What is a tentative statement about the natural world leading to deductions that can be tested? Option A, fact. Option B, theory. Option C, hypothesis. Option D, law. The correct answer is option C, hypothesis. A hypothesis is a proposed explanation made on the basis of limited evidence as a starting point for further investigation. Question 58. In science, what is a well-sustained explanation of some aspect of the natural world that can incorporate facts, laws, and tested hypotheses? Option A, observation. Option B, theory. Option C, hypothesis. Option D, opinion. The correct answer is option B, theory. A scientific theory is a comprehensive explanation that has been extensively tested and is supported by a large body of evidence. Question 59. What is a descriptive generalization about how some aspects of the natural world behaves under stated circumstances? Option A, hypothesis. Option B, law. Option C, variable. Option D, assumption. The correct answer is option B, law. A scientific law describes a repeated observation or relationship in nature, often expressed as a mathematical equation.